So when you're ready, Aaron. And we're live. So welcome back, everybody. My name is Martin Leahy. This is the Biophotonics and Imaging Summer School. I'm in Galway. I'm talking to Brian Wilson in Toronto at the Queen Margaret Medical Centre, I believe. And he has given us some lectures over yesterday and the previous days, which you have presumably viewed live or recorded. I see you've started asking questions. I don't know, Brian, if you want to say a few words first, or we go right into the questions.
I think we can go straight in and see where it takes us.
Okay, very good. So Ata Kazari in the Netherlands is talking about table of yesterday's lecture. It was at, yeah, I don't know if the guys can get this up, 4140 in the lecture yesterday. Accuracy was stated as a practical limitation of laser Doppler flowmetry. From a clinical point of view, to what extent should laser Doppler flowmetry be accurate?
That's a little difficult question because it's a little without particular context. I mean, I think in general, the accuracy that you need of any of these techniques is very dependent on what the particular clinical application is. So if you're looking at laser Doppler flowmetry, essentially to look at the adequacy of blood perfusion in a tissue, then it presumably makes a difference whether or not you just need a yes/no answer. In other words, is there blood flow, or whether you need to be able to get a handle on how much blood flow there is, in other words, to quantify it. So if you think about maybe the first case scenario where you have perhaps a surgical reconstruction. So let's say that you have a patient who has inadequate blood flow in the leg due to say a blockage in the femoral artery, and the surgeon goes in and removes that blockage, or tries to remove that blockage, and what they want to know at that stage is, well, do we have blood flow back in the lower limb? In that case, probably accuracy is not so critical because there may not be much ability of the clinician to juggle that parameter. But what's really important is whether or not blood supply has been reestablished. On the other hand, if you're looking at using assay to monitor the progression of skin burns, then you may, whether or not you need absolute accuracy is another issue, but you would certainly want to know that the measurement that you make on one day can be compared with the measurement on the following day and on the following day and the following day. So you want the relative accuracy to be high. And that may be partly a question of the actual technology, but also can be quite strongly dependent on how the technology is used. And so, for example, the care is not taken in the setup. And you are making the measurement at some different angle or a different distance from the tissue, then you may not get reliable results. So I think you have to define the question a little more specifically to know whether or not the device will be adequate for the purpose the clinician wants it for. But
I could probably make a couple of comments on. Yeah, I could probably make a couple of comments on it because I used to be substantially involved in this area, both through my PhD in a company called Oxford Optronics in the early days, and I think it's probably gone over to the salespeople now. So, yes. It may be really important to remind people that you don't get absolute values with laser Doppler flowmetry. You get relative values, which depend also on the optical properties of the tissue and the exact site that you use. So as Brian said, if you don't go back to the site and the angle and approach it the same way as you did previously, you will have issues. Almost always what you need to do is to have a relative measure of some sort. So you're at a measurement at the same site, either continuously. So, in the case of plastic surgery, as Brian said, Henrik Svensson did a lot of work where you keep the probe in the same place for several days. And if you do that, you could actually save, half of the failed flaps. And that sounds great, except the surgeons never really took it up. And you think, wow, this is saving half of the failed flaps. Why don't they take it up? And the point turns out to be that, that they're already, by their surgical skills, saving 90%, or 95%. And this could get them to 97 or 97.5%. But it just seemed that they really didn't want to spend more time on that. They wanted to focus on the 95% that they were already scoring, which they thought was very, very good. And, it did mean that you had to have very, very careful support staff like nurses and so on, who would keep this probe in position over long periods of time. So I would say relative measures, changing posture and things like that, so you can see what is it like before and after that stimulus, rather than pointing it and looking at a number and writing down the number and taking away your probe and bringing it back the next day, which I just don't think would work. So, okay, so Ming Lee and Lund.
Just comment a little more on that. It's worth remembering that in all of these technologies, we should clearly distinguish between accuracy and precision. Because with some of the technologies, it's possible to get tremendous precision. In other words, you can, if you're measuring a signal, you can measure that signal to several decimal places, if you like. But if you measure it to several decimal places, but it's wrong by a factor of 2, then you may not be achieving the required result. So be careful to distinguish between the accuracy of the measurement and the precision.
Yeah, and I suppose something to carry over from the previous session, we talked about the fact that, physicists and engineers have to get used to the idea that 10% accuracy or even 50% accuracy may be enough for a particular clinical situation if it answers the question.
Right.
But
right. And also,
we're used to.
I mean, just to tell a funny story on that. I mean, ultimately, if you have a physicist or an engineer in an operating theater or an endoscopy unit with a clinician, it's the clinician who calls the shot on whether or not they are satisfied with the result. And so you have to recognize that at some point clinical judgment becomes very critical, and you can't insist that your measurement is a correct measurement and therefore something specific should be done. It's the clinician's decision as to whether or not to do something with the measurement.
Right, very good. Meng Li at Lund comments that in your yesterday lecture you mentioned the biomarker cocktail contrast agents coated with different Raman active molecules. But by mixing all those agents, would there be cocktail effects, responses in the patient, if there's a negative effect in the mixing of the biomarkers? And secondly, as the biomarkers are made to target specific tumor or cancer cells, why not use it as a drug carrier to kill the tumor and cancers instead? Why is surgery needed?
Right. So let's take those two separately. So I guess the first question is really about, I was talking about the SIRS nanoparticles with multiplexed imaging, where you make a cocktail. One of the challenges of that technique, which we've found and have not entirely solved, is that if you use, say, antibodies as the surface coating for targeting to the tumor. And that antibody has to bind to a so-called antigens on the tumor cell surface. Imagine the tumor cell with a marker on the surface, and you have a nanoparticle coming in with an antibody that's going to link onto that surface. Although nanoparticles are small, they still have finite size, and therefore you do have the possibility that the more you attach the nanoparticles to the cell surface, which you want to do because you get more signal and therefore the measurement's more sensitive, the more space you're taking up on the cell surface. And so at some point, you reach a level where you're really saturating the binding sites. And for some particular biomarkers, there may be relatively few binding sites actually on the cell surface. And so that is not really chemical interference. It's really an interference due to the biomarker targeting. And the other quite difficult problem in that whole technology where you're trying to attach nanoparticles onto cell surfaces is that if we take a certain nanoparticle and decorate it with antibodies, antibodies are Y-shaped structures, and you want the Y to be sticking up from the surface of the nanoparticle, and it's actually quite difficult to get that to happen. So what can easily happen is that the antibody actually lies on the cell surface instead of sticking up from the cell surface. And of course, if it's lying on the cell surface, it's not going to bind to the tumor cells. And so those detailed chemical issues are actually pretty tricky and are not completely solved. So maybe, and I hope that sort of answers the first part of Lee's question. Well, what was the second part?
As biomarkers are made to target specific tumor or cancer cells, why not use them as the drug carrier to kill the tumor instead? Why do you need surgery?
Okay. Again, let me try to break that into a couple of different parts. There's certainly lots of nanoparticles are used to deliver drugs to cancer cells. And as I said yesterday, many, or not many, but quite a few chemotherapeutic drugs are actually delivered in liposomal nanoparticles. Some of those, but not all of them, are targeted to the tumor. In some cases, you rely on just the passive so-called EPR effect, the enhanced retention and permeability effect in tumors. But in some cases, you try to use something like an antibody or a peptide that targets the tumor. But it's certainly possible, and people do it all the time, to use just an antibody linked to the chemotherapeutic molecule for enhanced delivery. And so you can deliver drugs in that way without the use of nanoparticles. The advantage of nanoparticles is that in principle, they give you a much higher loading. So you can carry a lot more drug for a given number of binding sites on the cancer cell surface. And of course, chemotherapy, drug therapy, as I said right at the beginning, I think of the fourth lecture yesterday. Drug therapy is used as a primary therapy in only about 10% of cancer patients, and surgery is used in half of cancer patients. So I don't think you're going it's going to be a long time before we get to the stage where surgery is no longer used in cancer. Because think about it, if you have a large tumor, let's say you have a patient with a large tumor in the liver, there's a lot of cells in a gram of tumor. And so if you have a chunk of tumor that's 100 grams, that's a lot of cells. To kill those cells by drugs is pretty challenging, because the drug has to get into the tumor, has to be taken up, etc. And so in some ways it's much easier to, first of all, remove the bulk tumor, rely on surgery to take the bulk tumor away, if you can get to it safely, and then use the other therapies like radiotherapy, chemotherapy, immunotherapy to try to deal with the residual tumor. It's much easier for a drug or an immune modulator to kill 100,000 cells than it is to kill 100 million cells. And so you shouldn't think of chemo and surgery as alternative, but rather as complementary.
Right, but on that point, if I may ask, are there cases where you would want to use the drug or chemo first and then the surgery? And are there cases where you would go the other way around?
Yeah, for sure. Most cases where both surgery and chemo are used, I would say, I don't actually know numbers for this, but I would think it's a significant majority of patients have surgery first and then chemo. But it's fairly common also to do so-called surgical downstaging, where the tumor mass is too large for an easy or a safe surgical resection. And so often in that case, drugs will be given to try to shrink the tumor to so-called downstage it, so that it becomes surgically more viable.
Mm-hmm.
So both are used for kind of different purposes.
Would neoadjuvant therapy be one of those?
Yeah. Or sometimes neoadjuvant can also refer to chemotherapy post-surgery, but it can be chemotherapy pre-surgery as well.
Okay, I see the next question is from Adon Brannock in Galway, so I'll let Adon ask this question himself.
All right.
There you go.
In person.
Yes, thanks, Martin.
You might take the next few questions. Go ahead.
Okay, what endogenous optical biomarkers can be used for cancer detection in the liver besides hemoglobin and SO2? Could beta-carotene, bilirubin, or others be used?
Ooh, I'm guessing the question is in the context of tumors in the liver.
Yes.
Let me make a very general remark, and then try to get to the liver. I don't actually know that much about liver cancer, so you have to bear with me on that. But I mean, as a general comment, if you're going to use a tumor biomarker for targeting, whether it's for imaging or for treatment, you want the biomarker more highly expressed or in higher concentration in the tumor than in the host normal tissue. Okay. Negative contrast is very difficult to deal with. In imaging, if you have a biomarker that is downregulated or in lower concentration in the tumor than in the surrounding tissue, then you're looking for the absence of a signal in an image, and that's, from a information science point of view, much more difficult. And of course, if you're looking at a biomarker that you want to target some therapeutic agent to, then for sure that needs to be a positive biomarker. And so you can't really use a biomarker, or it's not usually very viable to use a biomarker that's in very high concentration in the host organ. And so if you take the ones that you mentioned of say bilirubin, that's in high endogenous concentration, and maybe it's the case, I don't actually know, that there's less bilirubin in a liver tumor than there is in liver. That may well be the case, but I'm not sure that you can do much with that information.
Okay.
Is that sort of reasonably answer your question?
Yes, that was.
Yeah.
Yeah. So we'll move on to the next question. What type of detectors and light delivery strategies are best for integrating DRS on small surgical tools? Are there any trade-offs or disadvantages due to minimization?
Sure. But there's always trade-offs. It doesn't matter whether you're talking about DRS with surgical tools or some other technology. I think for DRS, one of the advantages of spectroscopy compared to imaging is that you can do the sensing by optical fiber, and so the optical fibers can be as small as you like, down to a few microns in diameter, as long as you can get enough signal. And so in that case, the miniaturization challenge is usually reasonably straightforward. So there's multiple examples of incorporating fiber optics into surgical tools. If you want to do imaging, say in the surgical field, and you wanted to have some sort of imaging device integrated into a surgical tool, that's a lot more challenging in terms of miniaturization. I know that there are technologies, and in fact there's a nice project in Ireland on it, using extremely small cameras in the sort of millimeter diameter range with very high resolution. And I think those sort of technologies may well revolutionize what can be done in terms of integrating imaging directly onto surgical tools. Obviously you can buy a millimeter camera now, but it doesn't have good spatial resolution. So what you want is a 1 millimeter camera or less that's got a large number of pixels, so you get good resolution. So that technology, I think we're going to see in the next few years.
Yes, and just to follow up on that, would there be scope to use photoacoustic imaging to guide the tool, because you get a very strong signal from the metal?
Don't know if that's been done. I would worry about artifacts.
I'm talking about like guiding it from the surface, say like you would have a handheld probe.
Yeah.
Not to image on the tool itself.
Right. But you mean, sorry, just to clarify, you mean doing photoacoustic imaging in the tissue to localize where a surgical tool is?
Yeah.
Yeah. I guess I haven't thought about that. I guess you'd have to make the surgical tool very highly absorbing, I guess.
Yeah.
So you'd want a black surgical tool. And in that case, I guess it would work. I would be a little bit worried about artifacts in that situation, because it may be difficult to get to avoid having the signal being so large that you get sort of ghosting artifacts. I'm not sure how easy it is to do that.
Okay, thanks. So the next question is from Anonymous. What are the challenges in getting FDA approval for nanoparticles?
Oh, that's a very big question. So I mean, as I'm sure the questioner knows and everyone knows, there's lots of challenges in getting anything approved by the FDA or by any other government regulatory agent. And you know, it's good that there are many challenges, because we want it to be difficult to have unsafe medical products. For nanoparticles in particular, there's really a couple of different things to think about. The first is, and obviously we're mainly concerned about toxicity. Although I'll come back maybe in a moment and talk about efficacy. But if you look at toxicity, there's a couple of different sources of potential toxicity from nanoparticles. The first comes from the material of the nanoparticles themselves. So what are the nanoparticles actually made of? So I showed yesterday examples of, say, quantum dots, which are semiconductors, versus, say, our own porphyrosome nanoparticles, which are made of porphyrins and lipids, or say the X-ray radiodynamic therapy nanoparticles, which might incorporate scintillation nanocrystals. So obviously things like semiconductors and perhaps to a lesser extent something like cerium fluoride scintillation nanocrystals, there's a concern as to whether the actual material is toxic, because those are foreign molecules or foreign materials in the body, and so that's a major question around those types of nanoparticles, which is mostly the so-called hard nanoparticles that are fabricated from typically nanocrystal type materials. On the other hand, something like the porphyrosome nanoparticles that we developed, and there's lots of other examples, that's made from lipids and is made from porphyrins, both of which are naturally occurring molecules in the body. So we'd expect the actual chemical toxicity of those to be much less. And that we actually find to be the case. It's not to say that it's zero, because you know even water is toxic in a high enough concentration, but it's much less of a problem than with the inorganic hard materials. So that's the composition, the actual chemical constituents are involved. And the other aspect is whether or not there's toxicity from the very fact that you have a nanoparticle. And for example, I'll take this because it's very high in our own thoughts at the moment. When we take porphyrosomes, which are these lipid porphyrin nanoparticles, so there's no toxicity, no measurable toxicity from the material, and we give those nanoparticles to mice, we can give a huge amount of material, and in fact so much that we actually can't deliver anymore, because they aggregate in the syringe. And if we do that experiment in rabbits, it's the same. We can give as much as we like. If we do it in dogs, it's fairly easy to kill the dogs. And if we do it in non-human primates, it's fine. And so there's something about that particular species where it's not the material of the nanoparticle, it's the fact that there's nanoparticles. And it's related to upregulation of something called the complement system, which is a kind of almost an anaphylactic reaction or an allergic reaction, which is species dependent. So that the nanoparticles, the very fact that you're using nanoparticles can actually be problematic. So that's on the toxicity side of getting through FDA. So obviously you need all these, you need animal studies, toxicity studies in at least 2 species. One of the species must be a large species, and it's better if it can be a primate species, which raises lots of cost and efficacy and ethical issues. Then there's very strict protocols as to how you start with the first patient. So you have to start with so-called microdosing, which is at a level several orders of magnitude lower than the dose at which any effect has been seen in any species, and then you have very careful escalation protocols for how to increase to a therapeutic level. So that's on the toxicity side, but the other thing to remember is that the FDA for sure, and I think now probably almost all government agencies require that you also are concerned with efficacy. So you can't give a material, whether it's a nanoparticle or a drug, just because it's safe. If it's not effective for the purpose, then you're not allowed to use it. And there are examples of many examples actually of drugs that fail with the FDA, not because they're unsafe, but just because they're not effective. And so you have to think about that aspect as well.
Brian, if I can come in there, just on that topic, aren't there many lotions and potions that you can buy in your local so-called health shop that you can take, which are which don't have any proven efficacy?
Yes, they're not regulated drugs. So there's a difference between a regulated drug that has an FDA approval and an off-prescription substance that you may use. Of course, and the rationale for that is that those substances are not put through randomized clinical trials, etc., because there would be no economic viability to have that happen, and so one of the differences is that the non-regulated drugs cannot make medical claims. And so you can't buy something off the shelf at your health goods store that claims to cure cancer. That would be illegal for that claim to be made, and that product would be forced out of the stores. You can't make a medical.
Don't they claim that they improve your mental state or
yeah, of course,
do all kinds of other nefarious things that improve pain, back pain? I mean, some of them claim to cure almost everything.
Yes, yeah. So the wording is very difficult, and I think it's a matter of how much resources a government agency can put into going after people. I mean, if so, it's a fine line. But if I make some material in my kitchen and start selling it and say that it will cure people's headaches or reduce people's headaches, then it's not worth anyone's while going after me unless it turns out that I'm clearly doing harm to people because I'm selling them that substance that's harmful. And in that case, that'll be closed down. But there are so many of those types of preparation that you could never hope to regulate it. So it's just it's a practical issue as much as anything. You could claim that cornflakes cure hunger. It's perfectly true.
Yeah, Yeah, I do get what you're saying. We're probably going off topic, so I'll just leave it with this bit, which I think is probably you're always doing harm because you're taking on vulnerable people, and maybe we'll leave it at that. Back to Adon.
Okay, so we'll move on. The next question is from Martine Cooper in Amsterdam. They ask, in what size range can porrhizomes, porrhizomes be produced? Does their size influence their application?
Thanks, Martine. That's a good question. Yes, the size does influence. The standard porphyromes, where you take a porphyrin molecule and a lipid and conjugate them and they self-form, self-assemble in sort of the 70 to 100 nanometer range. Now you can change that by selecting different lipids. Because if the lipid is a more rigid lipid, then it will make smaller liposomal nanoparticles. But generally, in that sort of size range, for some purposes, that's a little large. It would be nice if there were perhaps more in the 10 nanometer range if you're going to target them, and so we've looked at that, and we can't really make porphyromes as small as that, but we can do a trick, which is to incorporate proteins, apoproteins, if you know the chemistry, into the material, and those proteins act almost like bands around the nanoparticle, if you like, compressing the nanoparticle and making them smaller. And those we can make. We call them mini porphyromes. We can make those in the sort of 10 to 20 nanometer range. And it turns out that those actually, for reasons that we don't really properly understand, are actually better PDT agents than the original porphyromes. They're not so good as photothermal agents or as photoacoustic agents, but they're actually better PDT agents, probably because they can more easily get into the tumor cells because of the smaller size. Because with photodynamic therapy, you really need to get the porphyrin into the cell. With photothermal therapy, it doesn't really have to get into the cells. As long as it's in the tumor, then you'll generate the heat, and you get thermal diffusion, which will incorporate, which will encompass all of the tumor cells. So it does look as if smaller porphyromes are better for photodynamic therapy. But to achieve that, we have to use this extra trick of putting a kind of a band of protein around them to compress them.
Okay, thanks for that, Brian. The next question is by Ashwini in LIU in Sweden. She asks, two-part question. If there are, what are those potential clinical uses of fluorescein over ICG other than vasculature imaging?
I don't particularly know of any. I mean, the advantage of fluorescein is it is activated at much shorter wavelength, and therefore the spatial resolution of fluorescein imaging is better than the spatial resolution of ICG imaging. But I don't know of other particular advantages.
Okay, we'll go to the second part. She asks, what is the trade-off between fluorescent signal stability and depth that a fluorophore can reveal?
Sorry, can you just read that to me again, please?
What is the trade-off between fluorescent signal stability and depth that a fluorophore can reveal?
I'm not sure what Ashwini is meaning by fluorescent stability. I'm guessing it may be referring to photo bleaching. So let me assume that is, and maybe Ashwini, you could send a follow-up question if that's not what you were referring to. So photo bleaching trade-off with depth. I'm not sure what the answer to that is in a general sense. I mean, obviously, if you're talking about, say, surgical guidance, then photo bleaching is always a limiting factor. In fact, that's one of the problems with ALA PP9 is that PP9 is relatively easy to photo bleach, and therefore, when you're doing fluorescence guided surgery, you have to be careful not to just have the operating rooms, operating theater lights blasting onto the tissue surface, because the PP9 will just disappear. I'm not sure that the bleaching is particularly related to the depth, the capability of depth imaging, but maybe Ashwini, you could clarify whether it's photo bleaching that you're referring to there.
Okay, so we'll see if she follows up. The next question is from Evelyn Granizo in Moscow. When using nanoparticles, once the signal is taken, are they completely eliminated by the body, or is it part absorbed?
Evelyn, that depends on the nanoparticle. So in the case of organic nanoparticles, to a large extent, those are metabolized. They are broken down in the body and are cleared either through the biliary system, through the gut, or in the urine. The inorganic nanoparticles may actually hang around for quite a long time. So that, for instance, it's known that gold nanoparticles tend to have a very long residency in tissue. So they don't really clear. There's an initial first pass of clearing, but those that are actually taken up into cells in the body tend to stay there for a long time. And so that long-term toxicity has been a bit of a question with some of the materials. So depends on the material, but as a general statement, the inorganic nanoparticles tend to stay in the body, are more difficult to be cleared than the organic nanoparticles, because the inorganic nanoparticles are more difficult to break down.
Okay.
And then, of course, there's also some concerns about toxic metabolic products. So just because, say, a nanoparticle breaks down in the body, it doesn't mean that it's necessarily safe. It might be that the breakdown products are actually more toxic than the original nanoparticles, and so that has to be looked at as well.
Yeah. Okay. Thanks for that. The next question is from anonymous user. When you design a contrast agent like a nanoparticle conjugated to an antibody or another nanoparticle, how can you make sure that the contrast agent is stable in vivo?
So there's kind of a series of steps with that's done with that. Of course, just going back to the previous question, you don't necessarily want it to be stable forever, because you would like it actually to break down and be cleared. But so the question is really, how can you make sure that it's stable for long enough to do whatever procedure you're trying to do. I mean, generally, when you're making nanoparticles and labeling them with antibodies, you first of all go through a series of stability tests just in the test tube before you get anywhere near to cells. And so you do shelf life experiments. So you make the nanoparticles, you have them in an environment where they should not disaggregate or break down, and you simply track them over time. And usually you have to show stability over at least several weeks, because if you think about a pharmaceutical, you have to think about how long it's going to take between the factory where it's made and getting into the patient. So you need to, first of all, just measure the simple shelf life. And there you have to worry about temperature and other environmental factors. It's often the case that you will measure the stability in serum, so in blood serum, because you want, if nothing else, to make sure that if you're giving an injection of the nanoparticles, that they don't rapidly break down in the bloodstream before they can get to the target, like the tumor. So there'll often be lifetime experiments in serum, and then you would go into animal models and track the stability as part of the whole tracking of the pharmacokinetics and the biodistribution. That's where it can get pretty tricky, actually, because depending on the nanoparticle, it may or may not be easy to actually determine whether or not it's still intact. So it's really, if you're looking at, say, a nanoparticle conjugated with an antibody, it's pretty difficult to have a way of testing whether or not the antibody and the nanoparticle are still associated once you've got it into tissue. You may be able to say what the nanoparticle is, but whether or not it still has the antibody attached is a different matter. So that tends to be not so much of an issue. However, the issue once you go in vivo is more a matter of whether the antibody is sticking to the nanoparticle for long enough that you get the tumor uptake that you want to achieve.
Okay, thanks, Brian. The next question is from Martine Cooper again. She asks, are there nanoparticles available which can be used for simultaneously destroying tumors with heat and radiation?
I mean, yes, in principle. For instance, just simple gold nanoparticles are a good example. So gold nanoparticles have been used for enhancing radiation dose. So as I mentioned yesterday, the idea, well, there are some different possible pathways, but one of the pathways is that the high atomic number of gold means that when there's a high absorption, high cross-section for X-rays, and you get secondary electrons produced, which then go on to produce the radiation killing. And so gold nanoparticles are used as radiation enhancers, but of course there's been a lot of interest in the biomedical optics nano community in using gold nanoparticles for photothermal therapy enhancement. So again, the very high optical cross-section of the gold nanoparticles means that if you, including out into the infrared, means that if you give in infrared illumination, you'll get an enhanced optical absorption and therefore an enhanced temperature increase in the target. So yeah, so gold nanoparticles are one example of a nanoparticle which intrinsically does both radiation enhancement and photothermal enhancement.
Okay, thanks, Brian. The next question is from Robert Stafford Williams. He's in UCL in the UK. In using exogenous fluorescent markers to aid image-guided tumor targeting, are there any issues surrounding overestimating target area, or is the tumor boundary typically well-defined enough for surgery/radiotherapy, etc.?
Another great question, Robert. Depends on the tumor. Some tumors are well circumscribed. In other words, they have a nice clean boundary, and in that situation, within the spatial resolution of the fluorescence imaging, the fluorescence boundary will accurately represent the actual tumor boundary. Where it gets much trickier is if the tumor is actually an infiltrative tumor, which is actually is the case for many tumors, where you may have a tumor mass, but there's also fingers, if you like, or extensions of tumor into the surrounding normal tissue. And if we look at the example of brain that I talked about yesterday, we know that you can identify a tumor mass. You can see a tumor mass on MRI, for example. But if you just take out that MRI tumor mass, then we know that we leave tumor cells behind. And we know that with fluorescence, there is an improvement in the ability to identify those, at least some portion of that infiltrating tumor. But it does mean that you have a challenge, because if you had, say, a brain tumor where there was, if you were all intelligent and knew that there was infiltrates of tumor going 2 centimeters away from the surgical bed, then, and you're making an image, then as you go away from the surgical bed boundary, you'll get a lower and lower and lower fluorescence signal. If you have a high enough fluorescence imaging intensity, you may be able to track that signal to quite a significant different distance, at least to 8 or 10 millimeters if you're working in near infrared. And the challenge that then poses to the surgeon is, well, where do I stop cutting here? And there isn't an absolute answer to that, except to say that usually if there is reasonable probability that fluorescence signal is coming from tumor. In other words, there's very little signal comes from the normal host tissue. Then that fluorescence signal says, yes, there is tumor there, and then the surgical decision becomes one of whether or not it can be safely removed. And so the surgical decision becomes one of safety rather than one of just chasing the weaker and weaker and weaker signal. And that's why in our own lab, we've been particularly interested in this ability to quantify, because at least in principle, that gives you some objective measure of whether or not a given level of fluorescence is clinically significant.
Okay, thanks, Brian. The next question is another one from Evelyn Granzio in Moscow. She asks, in just one fluorescent nanoparticle, is it possible to combine with isotopes in order to improve the detection, or must we use different nanoparticles for each technique?
There are some papers on, and this is been done particularly by a group in the Netherlands, and I think there are other groups making hybrid nanoparticles that are both fluorescent and radioactive. I mean, our own porphyromes are a good example of that, but certainly other groups have made nanoparticles that have both capabilities, and that's pretty interesting, actually, because if you think of the case of breast cancer, where this has been actually explored, one of the techniques that's used in breast cancer guidance in vivo, not to assess the lumpectomy specimen, but to actually look inside the breast, is to use scintillation counting. And so the patient is given radioactive material that has some tumor specificity, and then a scintillation probe is used to see if there's a signal, and that's used to guide the treatment. Now, the nice thing about that is that the penetration depth of the gamma rays from the isotope is quite large, and therefore you can detect the presence of tumor at quite a depth below the surgical surface. On the other hand, even if you were able to do imaging in that scenario, the spatial resolution would be very poor, and so now you come in with fluorescence, and you get the high resolution of fluorescence, even though you can't go so deep. So they're actually very nicely complementary. So there are, and a huge advantage of nanoparticles is you can make nanoparticles that have both of those functionalities and deliver them by the same targeting technique to the tumor. So you get both, you get the advantages of both of the techniques. So sure, that has been done.
Okay, thanks, Brian. Next question is from David McMahon in Tyndall here in Ireland. He asks, further to the moderator's PAT of surgical tools question, could we make the tool vibrate at ultrasound frequencies with motors to take out the laser middleman?
So I don't understand that. Say it again.
He wants to know, could you vibrate a surgical tool so that it would emit an ultrasound wave, and so you wouldn't have to use an optical source to excite the...
To guide the surgical tool, I guess.
Yeah, to visualize it or use it as a source of acoustic waves.
I have no idea whether you could do that. Let's think about it.
It might be safe to vibrate your tool like that.
That's a fascinating question. I mean, I guess I mean, if you just moved the tool, let's say there's the surgical blade. I mean, if you just moved the surgical blade backwards and forwards at acoustic frequency, assuming that you could drive it hard enough, and that's a question whether or not. I mean, could you make, in reality, could you make a surgical blade that's strong enough to be functional? Could you actually drive that at acoustic frequencies? And I'm not sure what the answer to that is. But if you could, then presumably that acoustic signal would be generated by the resulting compression of the tissue. So be a bit like ultrasound elastography. Or even OCT elastography, which I think David Sampson may have talked about. I don't really know the answer to that. It's an interesting idea. I just have no sense of. I don't have a good intuition as to whether or not that could actually be made to work. So it'd be interesting, I think, David, to see if there's any literature on that.
Yeah, it would. The next question is submitted by myself. What are some major unmet clinical needs or challenges for intraoperative surgical guidance?
Depends on the disease. So, I mean, I talk mainly about cancer because I work in a cancer hospital and most of what we do is cancer. But I showed you the example of cleaning up infected wounds, and an area that I didn't touch on at all was intravascular guidance. So if you look at the early days of biomedical optics back in the 80s, there was a lot of work done on trying to use optical techniques inside blood vessels to guide, whether or not you call it surgery is semantic, but to guide, for instance, treatment of blocked arteries. So quite a few groups worked on things like fluorescence spectroscopy, autofluorescence spectroscopy, and so on. And obviously, for intravascular imaging, you have the big advantage with optics that you can get down the channels. If you look at what's happened in that area, there's a couple of things that at least I know about. One is OCT. And I don't know if during the school anyone talked about intravascular OCT, but certainly the group at Mass General in Boston has done some really outstanding work on that, where they're able to take an OCT catheter into a coronary artery and make a complete reconstruction of the inside of the artery. Using OCT with very high resolution. One of the projects in our own group is using a scanning fiber endoscope. Now, I showed that yesterday in the context of the SIRS nanoparticle imaging. You remember the optical fiber that was spinning at very high, was undergoing a spiral motion at very high repetition rate that we use for imaging. So we've done that. We've used that inside blood vessels, and that gives you very, very high resolution real-time imaging inside a blood vessel. So an intervention, it's not quite a surgery, but an intervention that we've been looking at with that technology, which is a real unmet clinical need, is the placement of a stent. So you'll know that in many patients who have blockage in coronary arteries, one option is to cut out and replace the artery, but the other is to go in and expand using a metal stent, expand the artery so the diameter is maintained. And one of the big challenges there is how do you do that accurately? So typically it's done under X-ray fluoroscopic control. So you have real-time X-ray imaging going on. The catheter is coming into the heart, into the coronary artery, and you're trying to place a pretty small object very accurately at exactly the right location within that blocked artery. And you're doing that under fluoroscopic control, where you see a typical X-ray type images. So you kind of see the tip of the catheter, then you're trying to get it into the right position. So with the SFE technology, it looks as if it may be possible to actually see from inside. So you can actually see where you are inside the blood vessel and then put in the stent. And there is actually a company in Seattle, Veravante, that's developing that technology. So there's an unmet clinical need, not quite surgical, but certainly interventional need, where new optical techniques are going to, I think, be fairly revolutionary.
Great, thanks for that answer, Brian. I think we'll take a break now and reconvene at 7:30 or 7:30.
Okay, 25 minutes.
25 minutes, yes.
Okay, very good. Thanks, everyone.
Yeah, thanks so much, Wade.
Okay, so welcome back, everybody. My name is Martin Leahy, and we have Brian Wilson, I hope, online. And we're going to go back to questions in a moment. There you are, Brian.
Yes, hi everyone.
Very good. And Surya is going to take over asking questions. So away you go, Surya.
Thanks, Martin. So we'll start with Adon. So he has a question. What current methods exist for sensorizing biopsy needles to guide sampling location for cancer? Is there a preferred modality?
Yeah, so the question is there a way of guiding, when you take a biopsy, is there a way of guiding where the tip of the biopsy is? We've actually done quite a lot on that in collaboration with Fred LeBlanc in Montreal. So there are some papers on this. And particularly, we've looked at incorporating either Raman spectroscopy into the biopsy needle or quantitative fluorescence spectroscopy into the needle, or diffuse reflectance spectroscopy into the needle, or some different combinations of those three. The fluorescence and the Raman are primarily for the purpose of determining whether the tip of the biopsy needle is actually in the highest grade tumor. Because when you take a biopsy of a tumor, it's very important that you're able to take a piece of tissue that actually represents the highest grade, in other words, the most advanced state of the tumor, because the highest grade in the tumor is usually what determines the choice of treatment. So if you have a low-grade tumor, the treatment decision may be different, but is often different from a high-grade tumor. So it's very important to get that the highest grade tumor that's available. And of course, there's heterogeneity within tumors. So one region of a tumor may be relatively low grade and another region high grade. So Raman and fluorescence have been used primarily for that purpose. The diffuse reflectance, at least we have used mainly in the context of biopsying brain tumors for the purpose of making sure that there is not an adjacent significant blood vessel close to the tip of the biopsy needle. When you do biopsy of brain tumors, there's something like a 1 or 2% chance of killing the patient because you cut a significant blood vessel and you get bleeding in the brain that cannot be controlled. So that's a significant clinical problem. And so the idea that we had there was, if you imagine the biopsy needle inside the tissue, if there is a blood vessel close to this biopsy needle and the biopsy, the optical system is looking outward, say sideways, from the biopsy needle, then if you measure the diffuse reflection spectrum that you get, as the blood vessel gets closer and closer to the biopsy tip, you should see more and more of a hemoglobin absorption peak or signal. And so that information is used to determine whether or not there is a blood vessel within, I think we set the limit to something like 2 millimeters from the biopsy tip, because biopsy is usually done by suction. So you suck the tissue into the hollow needle, and so you don't want to suck that blood vessel into the needle. And the system that was built in Montreal, in fact, was multiple optical fibers all around the circumference of the biopsy needle, looking outwards. So this were all looking in different directions outwards. So you get like a 360-degree view of the tissue that's around the biopsy tip, so that you can make sure there's no blood vessels in any direction. So that's at least some of the work that we've been involved in for biopsy needle guidance.
Thanks, Brian. So next question is from Martine from Amsterdam. She asks, in what part or parts of the cell can we find most of the endogenous fluorophores? And does the amount of autofluorescence change with tissue type, or can you use it as a contrast agent for all tissue types?
Okay, I'm just writing down these questions, and there's autofluorescence for all tissues. So the first part, what part of the cell is the autofluorescence coming from? It depends on the excitation and emission wavelengths that you're looking at, because that would determine the fluorophores that you're detecting. So, for instance, if you use excitation and emission wavelengths that are sensitive to, say, fluorescence from collagen, then you will see you'll see the collagen distribution in the cell. If you look at the excitation and emission wavelengths that say report on or are excited by or that excite rather metabolic agents such as NADH, then of course you'll find those mainly in the parts of the cells that's undergoing metabolic processes such as respiration, for example, in the mitochondria. So it depends on the particular excitation and emission wavelength. So if you do fluorescence microscopy of cells and you change the excitation and the emission wavelengths or the detection wavelengths, you'll actually get different images because you're looking at different components of the cell. Are the autofluorescence characteristics of all tissues the same? No, they're not. They're quite variable, and there is no standard sort of autofluorescence background that you can use. Just to give you an example of that, when we did work originally on autofluorescence imaging in the GI tract, in the esophagus and in the colon, what we found, and we did this with fiber optics in an endoscope, what we found was that between patients, there was a huge variation in the autofluorescence signal, the spectra, from patient to patient. And actually, even worse than that, there was a huge variation from point to point in the same patient. And partly that's due to differences in, for example, the amount of blood that's in the particular location of the tissue, because the blood causes reabsorption of the fluorescence emission, and therefore that changes the measured fluorescence spectrum. But also the actual fluorophore content varies from point to point. So there is not a standard autofluorescence spectrum that you can use to represent tissue. You have to measure that spectrum in the particular tissue that you're interested in.
Aaron from Ireland asks, when designing a radiation therapy treatment using nanoparticles, are a large amount of nanoparticles needed for the treatment? And do a larger amount of nanoparticles pose a greater toxicity risk to the patient?
The answer to the second part of the question is yes. If the nanoparticles have significant toxicity within the range of doses or concentrations that you're likely to need. In terms of the first question, if we're looking at radiation enhancement, then as a simple statement, the more the better, because the higher the concentration, the greater the enhancement will be. That may be limited by toxicity, or it may be limited by simply your inability to deliver that amount of material. So that's one aspect, but the other aspect is that it may become self-defeating, because depending on the particular modality, the high absorption of the nanomaterial may actually start to provide some sort of self-shielding process, so that if you have a nanomaterial that has high absorption coefficient or high cross section for radiation, then the more you add, the more energy is absorbed, the more X-rays are absorbed, but of course the more the tissues and cells that are beyond a given position are shielded by the nanoparticles themselves. I mean, gold is used as a radiation shield, for instance, in radioactive treatments at the back of the eye. And so there is a limit to how much you can add. Which of these various factors becomes the rate limiting, the limiting step, is difficult to say. The other thing to bear in mind is cost. So, for example, when we were working on the CERS nanoparticles, and we had originally our primary aim, as I described yesterday, was for lung cancer theranostics. So one possibility was to deliver the nanoparticles intravenously, so that we give an intravenous injection of the nanoparticles, and that's how we tended to do it in the animal models, because it's easy. It's easier than trying to spray it on inside, say, a mouse's lung. That's pretty difficult. And so that works. But then one of our students, we asked to actually calculate how much, given how much we needed in the mice, how much would we need in patients, assuming a similar dose level. And so he went away and did the calculation and came back and said, well, it'll cost about $90,000 to inject one patient, because the cost of the material was such that at the doses that we needed, it became simply, even if ignoring questions of toxicity or biodistribution or whatever, you couldn't actually afford to do it. And so that's one of the reasons why we went back to saying, okay, we're going to have to apply these nanoparticles for that application. We're going to have to apply them topically. And so actually the cost of nanomaterials does become a significant issue, or can be a significant issue, if you're going to give systemic administration. So maybe if you're looking at that as a possible way of administering your nanoparticles, you might want to do that calculation early on to make sure that the treatment actually could be affordable.
Okay, thanks, Brian. The next question is from Julian. He thanks you for the instructive lectures, and he was wondering if you have any experience in using Raman to differentiate between LGG and HGG tumors in ex vivo or in vivo studies.
Okay. Yeah, so just to bring everyone else up to speed, what we're talking about here is low-grade glioma and high-grade glioma. High-grade glioma is the very malignant tumor where there's a very high risk of a very high death rate, unfortunately, because they're so aggressive. Low-grade tumors are slower growing and tend to be to go back to an earlier question that we had, tend to be well encapsulated. So it is actually possible to completely remove them at surgery, and it's actually possible to cure patients who have low-grade glioma by surgery alone. So differentiating between high-grade and low-grade is an important issue, and normally that's done by a mixture of imaging, preclinical imaging, and biopsy pathology that we just talked about. It's certainly true that again, if you look at the work that Fred LeBlanc has done on Raman guidance for brain tumor surgery, they've certainly been able to detect low-grade glioma in tumors in brain as part of surgery, and have published sensitivity and specificity values for high-grade and for low-grade. I don't actually know if they or anyone else has asked specifically the question whether or not there's a Raman spectral difference between low-grade and high-grade, a systematic difference. I imagine there might be, but I'm not sure if anyone's explicitly looked at that. It would be worthwhile looking at the LeBlanc papers, and I don't remember whether or not they actually published the actual Raman spectra in sufficient detail to allow you to see whether or not there would be a difference between them. But if that's not in the literature and is something you're interested in, I would suggest that you just contact Fred LeBlanc at École Polytechnique in Montreal and ask him whether or not he's actually looked at that. And if he hasn't, maybe he could, because he certainly has the raw data of the Raman spectra to be able to do that. So interesting question.
That's right. Next question is from Martine. She asks, are the optical properties such as refractive index or absorption coefficient of porphyromes similar to liposomes?
Well, certainly the absorption is different. I mean, that's because they've got this huge loading of porphyrins, and so the absorption coefficients are very different. In fact, the absorption coefficient of the porphyromes is really dominated by the absorption of the porphyrin, not by the absorption of the lipid. I don't actually know about the scattering properties. It's an interesting question. I don't think we have ever tried to measure the actual scattering coefficient spectra of porphyromes. Certainly something we could do, and it might be interesting to do. I would guess that it's going to also be different because the scattering is going to be dominated by the difference in refractive index. Well, I'll take that back. Maybe it's not so obvious. It is different. I'm guessing that the scattering is going to be dominated by the difference in refractive index between the lipid shell of the porphyromes and the aqueous core. Now, whether or not that would be impacted by the fact that these porphyrin molecules are stacked into the membrane, the lipid membrane, I'm not sure, actually. If we do the measurement, I'll let you know.
Okay, Robert from UK asks, in using cell targeting molecules, are there standard methods to optimize the drug light time and balance the buildup and clearance time of the molecule, or is it mainly a trial error during development?
I would say in general it's kind of educated trial and error, if I can put it that way. as I'm sure most people know, it's pretty difficult to predict the behavior of molecules in the body, except in some general ways. So if we look at some new targeting strategy of a particular molecule, really you need to do the experiments in a particular animal model system, for example, that you're interested in, or ultimately in patients. But you can do it in an informed way. So, for example, one of the photosensitizers that we originally used in prostate cancer treatments for PDT was so-called TUCAD. Some people may have heard of that, and it was known from the animal studies that actually it cleared that particular molecule, cleared extremely rapidly from the bloodstream. And so assuming that that was going to be the case in patients as well, and it turned out it was the case in patients, we actually started, when we gave the treatments, we actually started the light even before we had finished infusing the drug. So it took about 10 minutes to infuse the drug safely, and rather than just giving it as a rapid bolus. So we infused the drug over about 10 minutes, and then because the clearance was so fast, we actually started the light about 5 minutes after we started the drug treatment. Actually, we started the administration of the sensitizer. Now, if you look, for example, at a photosensitizer or some other drug that's targeted, say, with an antibody, you know in general that that's going to take a significant amount of time because you have to have the circulation into through the body. You then have to have uptake into the tumor because the molecule has to leave the vascular system, go through the wall of the blood vessels into the interstitial space in the tumor, and then it has to target the particular biomarker on the cell surface, and then it has to be incorporated through the membrane, through the cell membrane, into whatever is the target inside the cell. So you know that that's going to take hours. That's not a process that's going to happen in minutes. So in that case, if you're going to give an antibody targeted agent, you know that it's going to be multiple hours or maybe even some days to get the optimum time interval between giving that and giving the light. But ultimately, you just have to do the experiments. But it would be silly, for example, in that case, to do an experiment in which you gave the light 10 minutes after giving an antibody-based targeted photosensitizer or other drug, because you know that from general principles, you know that that's not going to be long enough to get that process, that multi-stage process taking place to get the drug into the target cells.
So Dr. Ashwin from India would like to know your views on the possibility of using gold nanoclusters for in vivo fluorescent imaging and photoacoustic imaging applications.
Mm-hmm. Yeah, I think there has been some work done. We haven't done any work ourselves on nanoclusters, but they are interesting materials. So I think it would be well worth looking at. We have been interested actually in, there is at least one paper in the literature looking at X-ray activation of metal nanoclusters, which is also interesting. So it's not, they're not materials that we've done any particular work with ourselves yet, although we're actually are trying to get a project going in that area. But yeah, have a look at the literature and see what's done. It certainly makes sense in principle.
Okay, the next question is, what is the state of robotic surgery and what are the challenges facing that?
It's pretty sophisticated. So, I mean, I guess the most the most sophisticated and widely used surgical robot system is a system from Intuitive, which is an American company that developed a robot with multiple articulated arms that can carry tools or other devices, including viewing devices, including cameras and endoscope and so on for keyhole surgery. That was originally developed, I believe, for cardiac applications, but the main application that it then was used for was in prostate cancer surgery, but now I know that that particular device is also being used for multiple other surgical applications. So they're pretty sophisticated. If you look at, and it's worth just, if you just Google Intuitive Surgical, you get an idea of how sophisticated it is. I mean, essentially the surgeon sits in a console, which can be remote from the patient. I mean, significantly remote from the patient, and gets a view, looks into a display setup and gets a view of what these cameras that are on the end of these arms are seeing inside the patient, and has various tools that he can manipulate to actually do the surgical procedure. So he will make a movement, and that movement will be translated into an equivalent movement of the surgical tool. So very sophisticated, mechanically, electromechanically, from an imaging point of view, and but ultimately that type of surgical robot is an extension of the human body, if you like. So it's like any other sort of human-controlled robotic device. The next stage of I think sophistication of surgical robots is going to be the incorporation of machine learning or artificial intelligence, so that some of those procedures can be done automatically without necessarily having the surgeon do the actual indirect control of the procedure. Obviously very challenging, but also very exciting, and could extend the capabilities, because after all, the human operator is also a mechanically constrained system. I mean, there's a limit to what you can do with your hands, whereas if the robotic devices that were incorporated were able to do procedures that could not be easily done by the human hand, even remotely, then that extends the capabilities of the robot. So lots of excitement, I think, in that area, and lots of opportunities for optical techniques to be used to help guide that and give you information for those types of procedures.
Okay, Mohammed from Dublin asks, could you please elaborate on why it is difficult to control thermal dose distribution with photothermal therapy? And he thanks you for your informative lectures.
Yeah, thank you. Yeah, Mohammed, I guess there's a trivial answer to that, and then a more complicated answer. I mean, the first answer is that once you generate heat, you can't control it. So if we put light into tissue, then say a photothermal treatment, then once we've generated that thermal jump, once we've got the temperature of the tissue going up, unless you add some form of active cooling, then the processes, the spatio-temporal distribution, then just depends on the thermal diffusion properties of the tissue. So one of the challenges in photothermal treatment is that you always get extension of the thermal effect beyond what you think may be the boundary. So if you look at photothermal treatments in different organs, you find that the thermal damage following after the treatment is finished, maybe hours afterwards, is in fact extends beyond the thermal damage that you think you're causing at the time of the treatment, and that's just because the heat continues just to spread. It's additionally complicated by the fact that blood flow acts as a heat sink in tissue, and so, for example, if you have if you're doing a treatment where you have an optical fiber in the tissue and you have a large blood vessel close by, then as you generate the heat from absorption of the light, this blood vessel can actually remove the heat locally and cause a cold spot. In other words, a spot of undertreatment in the region of that blood vessel. Even if it's not a large blood vessel, then the microvascular perfusion will carry heat away in a way that's really difficult to predict or to control, and it's further complicated by the fact that the thermal treatment itself may cause changes in the blood flow, and those changes can be either to increase the blood flow or to decrease the blood flow. So you know that if you put your hand in hot water, you get increased blood flow to your skin because it's a body reaction to try to remove the heat, and so you can get an initial increase in blood flow. But then if you go beyond a certain temperature, you start damaging the blood vessels and you'll get a decrease in blood flow. So it's a very dynamic process that is heavily dependent on the physiological response of the tissue to heat, and that's just very difficult to predict and very difficult to control.
Okay, thank you, Brian. So the next question is from Atta from Netherlands. She asks, what is the status of targeting and drug delivery employing microelectromechanical systems?
I don't know. Trying to think if I have anything intelligent to say on that question. Sorry, I really don't. I just don't know that area. So
No
I think it's better that I don't try to just speculate. But it's an interesting question. I just don't know the answer. Sorry.
Okay. Moving on. David from Ireland says, I have seen that your current research focuses on clinical translation of biomedical techniques, but what are your specialities within that scope? What technologies and techniques in particular do you aim to bring to the market?
Personally, I don't really have a specialty. That may be considered a weakness or a strength. I'm kind of interested in everything and special and expert in nothing. So the way our lab works really is that we work very much as a group of investigators, and we keep bouncing ideas off each other, and sometimes we collaborate. A collaborates with B on a project, and then maybe A collaborates with C on a project, etc. So I don't really personally have a specific focus for my own activity within our program, except to say that I guess my job nowadays is to try to drive the process towards the clinical translation and to facilitate that happening. Now, having said that, of course, there are things that I'm intellectually more interested in than other things at any particular time. So the thing that I'm most intellectually interested in at the moment is actually radiodynamic therapy, the X-ray activation of the photodynamic effect. So that's the thing that I'm currently finding intellectually most interesting and most challenging, but it's not necessarily what I'm spending most of my time on. So you asked a question specifically with the qualification of commercial translation. So I can tell you what I'm involved in there, which is I'm a co-founder of a company, and I mentioned it yesterday, IST, which is developing different techniques for for quantitative fluorescence imaging, but also for surgical navigation, particularly in neurosurgery. So that's taking some of my time, probably about a quarter of my time. And we are trying to create a company around radiodynamic therapy. That's much earlier stage, and is more challenging because it is so early stage that it's not entirely clear which direction to take that. And other than that, I personally consult for 3 or 4 other companies. For instance, I showed you Perimeter on the OCT for lumpectomy. That's one of the companies that I've been consulting for, and I'm consulting for another company at the moment is developing an optical method of detecting early Alzheimer's by looking at optical changes in the retina. That's not an area that I have specific biological expertise in, but I have, I guess, technological expertise in terms of how one might do that. So generally, I just spend my time doing things that I find interesting, and I guess I'm old enough that I can get away with that.
Hampus says, as you have raised this as a possible discussion point, why are we not using even shorter pulse lasers?
Good question. Well, it's a good question because I thought of it, but thank you for asking it back. So why are we not using even shorter pulse lasers? So I guess people know that there are shorter pulse lasers than are currently being used in the clinic. It's interesting, actually, if you look historically at the development of different generations of pulse lasers. So we started with the Ruby laser, which was a pulse laser. But then we rapidly went back to CW lasers with the gas lasers. And then over time, we got shorter and shorter pulses. And in fact, if you look at the evolution of laser technologies, it's about a factor of 10 to the 3 per decade. So from the invention of lasers in 1960, we're now at 2020, so 6 decades. So we've gone through, let's say, from CW down to now attosecond, so 10 to the minus 18 seconds. So it's about a factor of 1,000-fold shorter pulses every 10 years. And if you look at medical uses of lasers, you find that you have the same time track of evolution, but it's delayed by about 10 years, at least before it gets into the clinic. So a pulse laser, say microsecond pulse laser, developed at a particular time in history, you find that it's used widely in medical practice about 10 years after the original invention of that pulse length. So we've gotten to the point, as I talked about yesterday, where we're using pulses in the femtosecond range, and so the question is, well, what would we do with pulses that are at much shorter, at even shorter ranges, such as the attosecond range? And now, of course, if you know about attosecond laser science, you're in a domain where the interaction of attosecond pulses with materials is fairly different from the interaction of longer pulses, but the properties are not such that that's likely to lead, at least as far as I can see, to a significant medical application. I think in the life sciences domain, attosecond laser sources will be very useful, and already have shown themselves to be useful for fundamental biological research, and particular, for example, for looking at things like protein structure, where you can make the pulses so short that you can essentially you can sort of freeze the atoms in position in a molecule and get sort of the equivalent of X-ray crystallography, but without needing crystals. So that sort of very, very fundamental biological, biochemical information is only accessible by attosecond science. But I personally haven't seen anything where there's an obvious application where you would have an attosecond laser in a clinic. Now, having said that, you might say, well, couldn't you use an attosecond? You just said you can use attosecond lasers for looking at protein structure, so why couldn't you have an attosecond laser in an operating room? And when the surgeon takes out a piece of tissue, you analyze a protein structure using the attosecond laser, and I suppose that that's conceptually possible, but then you have to think about the other question, is whether that sort of laser will ever become ergonomic and affordable. So I think we're running out of direct clinical applications of shorter and shorter pulse lasers, but maybe I'm wrong, and actually, if I'm wrong on that, it might be very interesting. But I think we're reaching the limit of what we can usefully do, at least in the clinic, with shorter and shorter pulses.
Okay. Shanavas asks, if there are any optically active nanoparticles which have been used for applications in dentistry?
Not that I know of, but I may simply not know that. I don't really follow the dental application area very much, so I don't know. Certainly, nanomaterials are important for dental materials, but I don't know of any work that's been done specifically on optical nanoparticles for dentistry. But again, I probably wouldn't know that literature except accidentally.
So the next question is by Evelyn. She says, in uterine cancer, one of the ways to treat is by using fluorescence to visualize the cells and using laser to burn them. How can the person be sure that all fluorescent cells have been removed, or is this technique subject to operator errors?
Well, it goes back, I guess, to the discussion that we had earlier, that assuming that the fluorescence is only from the tumor cells, the question is how low a signal can you measure, and how low measurable signal would be actionable. So in other words, you have to think, well, what's the lowest that I could detect? And then even if I could detect lower than that, would I actually do something about it? So that question, I think, is the same for uterine cancer as it is for any other cancer. Obviously, technically you can make the fluorescence more and more sensitive. Becomes more and more expensive and may take longer to do, but ultimately the question is whether or not it's actionable. And the other thing that that's worth remembering in this is that if we're talking about an exogenous fluorophore, whether it's a molecular dye or some sort of nanoparticle-based system, as you get down to smaller and smaller amounts of residual tumor, the limiting factor, I think, mostly becomes signal to background rather than signal to noise. So it's the problem is not that you are unable to detect the very low level of fluorescence. The problem is that exogenous fluorescence signal becomes closer and closer to the autofluorescence tissue background, and so ultimately that's what limits many of the applications to try to get try to detect smaller and smaller amounts of tumor in a tissue background, because well, you might say, well, why can't we just subtract the autofluorescence background? But as I mentioned earlier, in the case of our measuring autofluorescence spectra in the GI tract, the autofluorescence spectra themselves can vary significantly from point to point. So ultimately, you'll reach a level of nanoparticle fluorescence where the variability and the uncertainty in the autofluorescence background actually overwhelms the signal that you're looking for. So that's kind of the ultimate limit, which is and that threshold is probably higher than the level that you can actually technically measure the fluorescence. So that becomes a limiting factor.
Okay, so the last question is from Robert from UCL. He first thanks you for your amazing lectures and thorough Q&A responses. And he asks, could we hear some of the key considerations in translating photomedicine tools and techniques into clinical use?
Well, that's actually what was the topic of what was supposed to be my sixth lecture. Lecture number 6 in the series, and we are going to try to record that and put it on the web. But I would also, Robert, refer you to a paper which Fred LeBlanc and Mike Germain and I wrote probably about 3 years ago. I think it was in Journal of Biomedical Optics, on challenges in translation of biophotonics, challenges of clinical translation of biophotonics. So that's represents much of our thinking around this subject. But just to sort of give you a bullet point list, I mean, the issues range from scientific and technical. Is your scientific concept of, your biophotonic scientific concept, is that a valid concept, and does it make sense in the setting of a possible clinical application? I always myself use the idea of deciding whether something is crazy but not stupid. So we can all think of really, really crazy ideas, but how many of them are also stupid ideas in the sense that they would never be able to translate. So you have those scientific questions, you have lots of technical questions. If it's a hardware, can you build it? Can you build it affordably? Can you build it ergonomically? I'll give you one example, which is in that 6th lecture of mine. I showed you the large camera system that we built for brain tumor fluorescence guidance, our first device that we built. And we thought that was a really neat piece of technology. And we thought, well, maybe we'll commercialize that. So we went to a local company, actually the company that makes the Canada arm in the International Space Station, because they had lots of expertise in electro-optical mechanical devices. And we showed them our machine, and they said, well, that's really a nice machine, guys, but you should realize that it is unmanufacturable. And we said, what do you mean? That's not even a word. You just made that up. What do you mean unmanufacturable? And they said that the design that you've done, you can make a one-off, but you could never manufacture it in quantity. And there were particular technical reasons to do with the fact that we had to slightly angle all the optical elements because we needed to get rid of specular reflection from the optical surfaces. So lots of technical issues, clinical issues, related to the clinical trial design and whether or not there's a clinical utility. And let me give you another example of that. When the original fluorescence endoscope was brought out, it was brought out for mainly in the lung. And so that fluorescence endoscope was commercialized by a company called Zilix in Vancouver in Canada that no longer exists. And what happened was that they brought out that device. It went through clinical trials. It was shown that you could detect very early, even precancerous lesions in the lung. And so they did large clinical trials, and they got approval from the FDA for the device. And so they started manufacturing the device in larger quantities, and no one would buy it. And the reason that no one would buy it was that at the time, there was no way for a general respirologist, and that was the main customers, there was no way, they had no method of actually treating patients with these very early pre-malignant lesions. So there was no point in detecting them because there was no treatment. And as a result of that, that company actually went under. Terrific technology, a great application, but actually one missing element was that it was not a valuable piece of technology at that time because of other aspects of the clinical situation. So, just give you that as an example of the sort of thing that can happen. And then, of course, you have to get through the FDA, to be able to scale up manufacturing, etc. And that's on the instrumentation side. On the nanoparticle side, there's a whole range of different problems. Again, if I take the example of the porphosomes from our lab, that invention was made in 2010, and we have not yet put it into a patient. So 10 years later and about $2 million of research grants later, we still haven't gotten that into a patient. And that's a long time and a lot of money. And although we've generated a lot of scientific papers in that and feel pretty proud of what we've done, nevertheless, we still haven't got to the point of actual clinical translation. So it's not easy.
Thanks a lot, Brian. So that was the last question. And thank you. Atta says thank you for this informative session, and he's learned a lot.
Good.
So thank you so much, Brian. I'll now hand over to Martin.
Okay. Well, thank you, and thanks to everyone who posted questions and who listened to the questions. I just wanted to say that it would have been very nice if we'd been able to be together and having these question and answers over a coffee or over some nice Irish food or over some nice Irish beer like Guinness. But I guess we'll have to wait until the next summer school for that to happen. But I enjoyed the Q&A sessions anyway. So thanks everyone for participating.
Thanks all. I don't know if you can hear me. My system is playing up a little bit, but can you hear me?
Yes.
Good. So this is Martin Leahy from Ireland. You were listening to Soraya James and Adon Brannock asking questions of Professor Brian Wilson from the Princess Margaret Centre in Toronto. And thank you so much, Brian, for all those wonderful answers. If I can ask you to make one short comment, which is on therapy, because I know that many of us get told that we're not doing enough on the therapeutic side, where 75% of the demand is and about 25% of the effort is. So in terms of standards and protocols, I know you've been involved in writing some of these in the past. Are these, do you think, well documented and easily accessible, and can people pick them up and go with those?
Mark, do you mean clinical trial protocols?
No, I'm not talking about trials. I'm talking about protocols for actual therapy. So photodynamic therapy, for example. I think you've been involved in writing some of the.
Oh, you mean how it's actually done?
Yeah.
Once it's proved. I would say, as a general statement, that this includes in PDT, at least for cancer, that's not as well developed as it should be, and that actually might be one of a limiting factor. I mean, mostly what happens in medicine, and including, but maybe particularly therapeutic medicine, is that once a procedure is approved, that then becomes part of the medical training processes for the particular specialist physicians or generalist physicians who are going to use it. So that gets incorporated into their training, and so the whole details of how the procedure is done becomes built in to that process. That's not nearly as well developed, I would say, for phototherapeutics as it should be. And part of the reason for that, only part, but part of the reason is that in a way, photodynamic therapy and the other phototherapies can be used for far too many things. And so there's far too many specialists can use them. So PDT can be used by a urologist, or by a head and neck surgeon, or by a neurosurgeon, or by a GI endoscopist, or by a dermatologist, or by an ophthalmologist, etc. And all of those different specialties have their own requirements and processes for training, for procedures. So there isn't a common training or protocol for PDT that straddles all of those. So that's actually has been a bit of a hindrance. So I would say in general that that information is not as available as you would like it to be.
Right, opportunity there for somebody to rectify that, I think. So Brian Wilson in Toronto, and to everybody who's participated in the question and answer session this week, thank you so much to all the team who asked questions, and especially to the people behind the scenes. Aaron Croak and Rajib is there today, but we've had many others on the team during the week. That ends our question and answer session for the Biophotonics and Imaging Summer School from Galway. But you will be able to view the videos and these questions and answers for the foreseeable future. And of course, many of the professors have kindly offered that you could make contact with them if you felt that you could maybe gain more from a further clarification or further question. They've been very, very generous with their time. So thank you for that, Brian. I know that Brian has also looked at some of the electronic posters, and we're hoping that everybody will have a look at those over the next day or so, and we will report back to you. I am hoping to present prizes on that, so do keep posted on that. Thank you all for participating. You are the people who made it successful. We wanted to access as many continents and as many people with bandwidth or caring or jobs or travel difficulties as possible, and I think we've achieved that with your help. So thank you all so much.
Okay. Thank you, Brian. Thanks for doing that, Brian.
You're very welcome. Okay. Thanks,
Good.
Brian.
Bye now. Bye, everyone.